begin by thanking the organisers for inviting me. I've very much enjoyed the conference so far. It's also my first trip to Prague, so I've also had time to look around the city as well, so that's been great as well. Thank you all, especially. Following last night's dinner and dance, I'm the first speaker, so I'm following Czech music and dancing. I won't be as exciting, I'm afraid, but thank you all for getting up early and turning up. I'm going to start my talk with a demonstration. How many of you know what this is? I'll give you a picture on the screen. Right, okay. Sun timer, okay? But for my talk, it's not a sun timer. This is a 10,000 to 1 scale. Believe me, have a supply of particles. The particles are delivered. I might put a target. Cells. We're delivering those particles. Okay? And this is sedimentation driven. You can see the idea. And of course, in this experiment, we know what's going to happen. I'll tell you the number on the top here is 3 minutes. We know what's going to happen in 3 minutes' time. Okay? And it's obvious that we have a finite reservoir of particles. This sand after some time is going to run out. And really the key message of what I want to say today is many of the inputs do are like this. A finite reservoir of nanoparticles. We have to take account of that fact when we try to understand the response of biological systems, potential toxicity of the particles. What I'm going to talk about is dose-response studies in this sort of picture of a finite supply of particles. So I don't think that's all. So I've started off here. If I go back and pick up at standard textbooks or whatever on pharmacokinetics, I can pick out 2 sort of limiting or model situations. Okay, I might deliver the toxin or the drug in some kind of bolus delivery and injection where the drug, or in our case, the nanoparticle, is delivered in a pulse form. Okay, so it's a sort of impulse, what I would call as an engineer, an impulse response. I have a finite, short duration of delivery, and then over time I'll look at the response of the organism to that delivery. At the other end, we might have a situation where we have continuous delivery, so under some kind of infusion. And now what we would be doing is trying to get a steady state level of the drug or the nanoparticle within the organism, and again over time we're studying that response. So this I might call an equilibrium response. And I think some of the issues we have in nanotox is that we don't have this situation and we don't have this situation. We have some kind of mix. We're at some way halfway house between these two, and that brings in complications. The other thing, of course, in vivo is there's direct delivery of the drug to the organism. And although obviously you worry about what happens to the drug once it's in the animal, you don't have to worry about is the drug getting to the animal. You know it's directly injected into the organism. Again, obviously in vitro systems, we don't have that, and we have to worry about how the particle actually gets to the cell to begin with before we then try and understand the dynamics of interaction with the biology. So if I move on to looking at in vitro, and this is primarily what my talk's about, is trying to understand nanoparticle assays in vitro, and also trying to see where we can get some standardization or some referencing. So very much ties back into what Kenneth Dawson was saying on Wednesday. Are there ways, are there standards, are there approaches that we can adopt, to try and get some agreement across groups, across the community? So the first point I'm trying to make in this picture here is here's our cell. We have our nanoparticles, which will be delivered to that cell via a host of different processes. Diffusion, sedimentation, and we've heard a lot this week again about the complexities of agglomeration and some nice talks yesterday about the time dynamics of that agglomeration as well. So there's a whole lot of stuff going in here, and eventually we get contact with the cell and internalization. But what I want to emphasize here is this stage of the whole process is an exposure. And the dose that we need to consider when we want to look at a dose-response curve, the dose is in the cell. And so when we use the word concentration, and this is one of the key points I want to make, concentration doesn't really, concentration only describes the exposure, okay? The dose, the key thing we want to know, that the thing that influences the biology cannot be described by concentration. And my key message is that we have to consider dose as number of particles per cell when we're dealing with nanoparticulates. And so to come back to what I had on the previous slide, what do we have when we do a nanoparticle experiment and a nanoparticle toxicity assay? Well, we have some extended delivery. So when we incubate cells with particles, we don't have a direct delivery of the dose. It's an extended delivery over many hours or perhaps even many days. And importantly, that arrival, that delivery of the nanoparticles overlaps with the biological response. Okay, so we have this merging of delivery dynamics and biological response dynamics. And again, that makes the situation more complex than if we have a nice clean delivery and then sometime later some biological response. Okay, this is just to show this isn't just a sort of pet thing of mine. You know, there is obviously a recognized need in this area to try and get better standardization. And again, Kenneth mentioned on Wednesday the series of editorials that have come out in Nature Nanotechnology calling for efforts to try and standardize the measurements. So this is, I think it's last September, is it? And here I've just taken a section from the editorial and just highlighted in red, you know, some of the important points. But basically, when we're looking at in vitro studies, typically, and I will be talking about this as well, where we have a system where we have adherent cells on the bottom of some kind of well or plate, and we're looking at the delivery of a nanoparticles to that cell layer, and then there are lots of misconceptions, inconsistent results, etc. And so this is sort of background of what I'm trying to talk to. So what I put up here is a schematic that shows the steps that we might consider in terms of initial exposure of an agent to the cells, through its delivery, interaction with the biology, and then eventually some response, okay, cell viability or other measures of the biology. And in this week we've heard lots about things like the delivery, the protein corona and the importance of that, diffusion, sedimentation, and the whole dynamics of mobility of particles in solution. And then obviously, especially if you're biologists, what are the mechanisms for cell death? What is the biological interactions, the biological responses? My point of view is focused way up here, right at the start. Okay, so what I'm talking about and what my work's about is trying to understand and quantify accurately the dose. And so for me, that stuff in the middle, I don't tend to worry about this, okay? I'm not saying this isn't important, it's extremely important, but we all have to have our focus somewhere, and my focus here is here, right at the start of all of this program. And so my approach, and the approach in our group, is to say, okay, I have a dose, I have some quantity of nanoparticles I'm going to deliver to a cell, and that's going to induce some cellular response. Okay? And in the data I'll show you today, that cellular response is cell death. And everything that happens in the middle, I treat as a black box. Okay? So this very much is part of my background. I'm a physicist by training, and I now work in an engineering department. And I noticed the title of this session today is about systems approaches. This, if you like, is systems analysis as understood by engineers. Okay? So in engineering textbooks, if you look up systems, basically what that means is you have a complex system, and instead of trying to understand the interactions of every component within that complex system, you wrap it all up into the black box. And the way you then study the system is you have inputs and you have outputs. And so in this case, our input is the dose, our output is the cellular response, and what we do is we have some mathematical function, represented by F here, and this mathematical function, this transfer function, is going to tell me, if I know my input, my dose, what my output is, my response. Okay. And of course, in this case, this black box function is none other than a dose-response curve, which I'm sure you're all familiar with. And so, if I can measure my dose, I can measure my response. The form of this response function, this is F here. And the form of this function obviously allows me to relate dose to response, but also the shape of this mathematical function does tell me something about the processes occurring inside my black box. Okay, so although I don't look directly at biological mechanism, the mathematical form of dose response does indirectly give me information about what's happening through all of the nanoparticle cell interactions. Okay, so to come back to my fixed reservoir. So what is happening when we do one of these experiments, these in vitro experiments? Well, I think what's commonly assumed, even if it's not appreciated that we assume this, but if you read papers, I think there is this underpinning assumption, is that the number of nanoparticles, so ND here it refers to the dose of nanoparticles I'm going to deliver to the cell, but the dose of nanoparticles is proportional to the concentration, the volume, the rate at which I transfer those particles. So here lambda is the average rate of transfer of my particles from solution to the cell, and time. So if I have a higher concentration, I'm going to expect a higher dose, obviously, and if I have a longer time for my assay, again, I would expect a higher dose. And the other factor in all of this, we can consider the supply of particles and the dynamics of the particles. The other side to the dose-response curve, the shape of that dose-response curve, is of course related to biological variability or heterogeneity. And so obviously in a dose response, what are we doing? We're looking at the response of the least susceptible, the drug-resistant cells, and the susceptible cells, and looking at that response across that whole variation in susceptibility of our biological population. So this, I think, is what's assumed. The problem here is, as I've said, we don't have a continual infinite reservoir of particles. And so over time, this effective concentration is going to change. And we can model that, or we can start to understand that, with some fairly straightforward maths, some fairly simple maths. So what I've done here is just to look at the rate of depletion of my reservoir. Okay? So this equation is just saying, over time, how fast do my particles move from my supply to my target? And so the rate of change is just proportional to the number of particles I have. So NR here is the number of nanoparticles in our reservoir or in solution, times the average transfer rate lambda. And so that's fairly easy to solve, and you get this exponential decay in the reservoir population. So over time, this is going to deplete the reservoir. And if I then look at, instead of what's leaving this reservoir, what's landing on the cells, what's the uptake into the cells, then I get this expression. Which is a very nice, simple expression where the dose, the particle number dose, is going to be proportional to concentration times volume. So that's my reservoir total. So C times V in this is the size of that initial reservoir that I'm using. And then this 1 minus exponential term, and this accounts for the loss over time of that reservoir supply. So immediately, if I look at this, this immediately tells me that this straightforward linear assumption where more time, longer the assay, the more dose, obviously is going to break down. So already we have to be wary when we do nanoparticle assays of the time aspect of the assay. Okay, so you might think at this stage, okay, that's all very well. Yes, you have a finite reservoir, but if I have a finite reservoir, if it's a big enough reservoir, I'm not going to worry about a few particles missing from it. So what I want to present here is a short study where we've tried to assess actually how many particles we get per cell. Okay, what's the particle number transfer in a typical dose-response assay? And so the system we're using, these are adherent osteosarcoma cell line, U2OS. We're using commercially available quantum dots, cadmium telluride, and these are receptor targeted. And what we do is we have a 1 hour exposure time of cell osteoparticles. And the results I'll show you here are for a 10 nanomolar concentration in 2 mil of volume. And so what we've tried to do is get absolute quantification in terms of particle number per cell. And to do that, obviously, we need to go to some imaging regime where we can actually see particles. And so we start with transmission electron microscopy. So the images here, I'm not sure how clear they might be to some of you, but this is a panel of images. Here's a section, a microtome section of a cell after loading, and then these are individual vesicles, lysosomes, in which you can see black spots. These are the quantum dots. And what we do is we've imaged 300 individual vesicles over a number of cell sections from a number of different cells. And what we can do then is form a histogram, frequency histogram of number of particles per vesicle. And so the red circles on this diagram are the data from the 300 TEM sections. And then what we do is we take a probabilistic approach. So we can fit that probability function. In this case, a bi-exponential mathematical term fits rather nicely the results, the experiment. And what we can do, once we have that fitting function, we can then randomly sample that function and artificially in the computer generate numbers of particles per vesicle. So the blue bars there are an in silico experiment, if you like, using this distribution, randomly sampling that distribution to produce a profile of particle number per vesicle over, in this case now, 100,000 vesicles. So the reason we're taking this kind of approach is, in the end, what we'd like to model is particle uptake over 10,000 cells. We want to understand nanoparticle dynamics over large populations of cells. Obviously, our colleagues, these images are taken by colleagues in Leeds University. They're very patient in taking 300 sections for us. If I start to say I want you to study 10,000 cells and multiple sections in the cell, obviously they're not going to be able to do that. And so by taking this probabilistic descriptor, which then we can randomly sample, it allows us to generate in silico as many mimic cells as we wish. Okay, so that gives us the number of particles per vesicle. The next thing we have to worry about is how many nanoparticle loaded lysosomes do we get per cell. And so to do this, we use imaging cytometry. So this is a really nice machine. It's from a company called Amnis. Basically, it's flow cytometer, but it has a camera instead of a photomultiplier tube. Okay, so you can rapidly analyze many thousands of cells in a flow system, but you get an image for each cell. And so here I've shown the fluorescence channel that picks up these quantum dot fluorescence, overlaid with a bright field. And so you can see what this now allows us to do through automated image analysis, cell by cell. We can actually count the number of loaded vesicles. And again, we can plot out a frequency distribution for that. And again, we can fit that with some probability distribution function. So again, the circles here are measurements from 5,000 cells from the experiment. The black line, which is the fit to that, is this probability distribution function. And then the blue bars are us randomly sampling in silico this distribution to create a modeled population of 10,000 cells. So now what I can do is I can create a model in which I can faithfully represent the distribution of nanoparticle loaded vesicles per cell, and I can faithfully reproduce the distribution of particles per vesicle in each of those vesicles in each of those cells. So that's all very well, and we're nearly there, but there's one final step. The problem I have here is these are sections, okay? So these particle numbers I have here, this is not the total number of particles per vesicle, it's the total number of particles in a 100 nanometer section of a vesicle. So I need to try and find a way of calibrating from a 2D slice to the whole three-dimensional vesicle or vesicle. And to do that, our colleagues in Leeds have have access to a really nice instrument, which is an electron microscope in which the ultramicrotome is situated inside the instrument. Okay. So in this instrument, what you do is you have your cell section, your cell block, embedded cell block, and then the instrument slices off section by section and then images the remaining block. Okay, so it's like a loaf of sliced bread. You image, take a slice off, image, take a slice off. And so what that allows you to do is take stacks of 100 nanometer sections, and because it's within the instrument, because you're imaging the remaining of the block, not the actual section that you've cut off, you can correlate all of those sections to reproduce a three-dimensional representation. So this is a 3D image of a whole cell. You can see particles here around the outside of the cell, and then as we section through, we can see each section and count the particles in each section. And so what this allows me to do finally is to calibrate the percentage of particles within a single section to the total number in the whole three-dimensional object. Okay, so this gives me my calibration factor, multiplying factor, to multiply from number of particles per section to total number of particles. Okay, so with all of that, we're now finally in a position to predict the total number of particles per cell over a large cell population. And so that's what I show here. So now this is a fluorescence. The red line is a fluorescence intensity histogram from a flow system. So this is now for 10,000 cells. And you see that range in fluorescence due to the different numbers of particles and the different number of loaded vesicles. And then the black line is a fit to that, where the black line is a prediction from random sampling of these various statistical distributions I have here. So you can see we have a really nice fit, and then what we can also do now, obviously, is calibrate that fluorescence histogram to particles per cell, because we've done all the counting through these processes. And so the outcome of all of this tells you that in this system, we've got something close to 2 million particles per cell. And if you consider that in a bit more detail, if I have 10 million, typically I might have, a million cells per mil, a million particles per cell. So that gives you 10 to the 12 particles loaded into cells. And if you're dealing with nanomolar concentrations and milliliter volumes, this is going to lead to total depletion of the available particles. And so this idea of a finite reservoir is important, because if you look at how many particles typically are loaded into cells and the typical volumes that we use in assays, typical number of cells, then basically we're going to run out of supply of particles. And the toxicity response, you see, is heavily dictated by this limitation of particle supply. Just to show that this isn't just something about the particular cell line or the particular particles that we use, here are some references from the literature, different types of nanoparticles, different cell types, different measurement techniques. There's a range here, but you can see the idea that many, many thousands, if not millions of particles get loaded per cell. You can see that in a lot of systems. So personally, I'm quite comfortable with the idea that we are dealing with 10 to the 5, 10 to the 6, even 10 to the 7 particles per cell when we typically do these assays. Okay, so let me come now to some data, some dose response data. And so what we've done is we've tried to find what I would call a model system to do a toxicity assay. And what I mean by model system is this is a dose response assay which is deliberately constructed to investigate the ramifications of finite supply of particles. Okay, so I'm not directly interested in the toxicity of this agent or the response of a particular cell population. This is constructed deliberately just to look at these dynamics of dose and how particle number and the temporal change in that affects the response in the cell. So what we use is PEI-coated quantum dots. So these are cationic nanoparticles with a zeta potential somewhere close to 30 millivolts. And the reason we use these is that these interact with the cell. They bind to the cell membrane. And you can see here in an electron microscope, this is a fluorescence picture. You can see the fluorescence from the quantum dots. And what's nice about this system is they tend to bind to the cell membrane, and then the cell death is fairly direct in the sense that it follows direct membrane disruption of the cellular membrane. And for us, what that means is it allows us to isolate the dynamics of the delivery of the nanoparticle from downstream biology. Because that death process is fairly direct and fairly immediate, it allows me to study dose in isolation from confounding effects of biological interactions. And so details of the Q dots and etc. you can find in this publication. So that's the system, and the results I'll show you here are with fibroblast cell line, and this is the particle system. How are we going to measure? So we're going to measure a dose response. So here's a typical particle dose versus percentage cell death dose response curve. But what we'd like to do is not do this in a standard dose response curve. And because for us, this has some limitations. Okay? Firstly, obviously, this is an endpoint measurement, and we're interested in dynamics. And so, obviously, this is just the end of a process, not the process itself. And so it's very difficult to say much about a process if you're just looking at the end result. And the other thing is, a dose response curve is a mean response over many, many, many thousands of cells. It's a population level measurement. And again, what we're interested in is trying to look at how this dose response curve relates to individual cell responses, the biological variability. And so we'd like to measure at individual cell level, and we'd like to measure a time resolved in a time resolved way so that we can study the process and the evolution of this dose response curve. So to do that, we use microscopy, time resolved microscopy, and in particular we use a high throughput system. It's a GE Healthcare in-cell analyzer. So this is basically an automated microscope which allows us to take images. In the data I'll show you one image every hour over multiple fields of view. And so if I can get this video to work. So here's a typical field of cells at the start of the assay. And what I'll show you in the movie is a 24-hour time lapse, one image per hour. And what we're using here is an agent called DRAC7. This is a membrane impermeable agent. And so this works rather nicely. We can incubate our cells with this DRAC7 agent. And then as the cells go through the death process, obviously we'll see influx of this fluorophore and labeling of the nucleus. Okay, so it's a nuclear label that we see upon cell death. So let's see if I can get this movie to play. Yeah, good. Oh no. Okay. Great. Okay, you're not going to see my movie, but okay. Well, if the movie played, if you really want to see it, obviously come and find me a coffee. I'll show you on the laptop. So obviously, what you see over time is the cells starting to round up as they go through death, and you see red spots appearing, which are the counts of this DRAC7 agent. The important point being is what you get is nice, clear, red labeled nuclei. You can then automate the image analysis, so you can automatically detect cell death and the point of cell death. And you need to do that in an automated way because we're doing 81 frames at each time point and one time point every 24 hours. So in the end, what we end up with is a population of something just over 3,000 cells. Okay. So here's the results at 24 hours at the end of the assay. Here's our exposure, nanomolar concentration range, percentage cell death. But of course, as I've been saying, we've done this in a time resolved way, so we have a lot more information than just this endpoint. And so what we have is here percentage cell death over time, so we can see the evolution of the death across the cell population over time, and obviously we can do that over our full range of exposure concentration. Okay, so what I want to talk a bit about now is what drives the shape of these profiles, what drives this whole response and this temporal dynamic. So what we're doing here effectively is we start off, what we've got is some biological variability. So this represents some distribution in susceptibility. And what we do in the experiment is we expose those cells to a dose, and depending on the concentration or the time, as the dose increases, obviously the most susceptible cells we see die first, and then over time, or as I say, for varying concentrations, we scan across this profile, killing cells as we go until we finally, if we come right across a profile, even the most drug-resistant cells will succumb to the agent, the toxic agent. And when we do these experiments with these finite reservoirs, what effectively we're doing is we're scanning across this profile up to the point of the concentration times our volume, the total number of particles we have in our experiment. And that might not be enough to scan across that whole profile. And what we do then is that integrated area becomes our cumulative cell death, and so this gives us our cumulative response, which is the dose-response curve. So this is what we measure, various points along this curve. Okay, so I've got a few minutes left, so I'll push on. So to model this, to try and fit this data, what I basically need to know is this biological heterogeneity. And this is the other nice thing about having a model system. We've created our assay system here in that we can directly measure the biological variability because the heterogeneity here arises directly from cell area. Okay? So the agent, the nanoparticles, are labeling or landing on the cell membrane, and these are plots of the fluorescence from a single cell over time. So each of these curves is that increase in nanoparticle dose in a single cell over time. As you can see here, cells dying later on after 24 hours, cells dying earlier on in the assay. And so that different response of the cells is due to this different uptake rate of the particles, and that uptake rate is directly proportional to cell area. So the variability here is the cell size, with larger cells basically physically having a larger cross-sectional area, and so they're more likely to come into contact with nanoparticles. And so to model this data, I can take the measured distribution of cell area. I get that from my microscopy. So here's my biological heterogeneity profile. From the model of a finite charge reservoir, carrier reservoir, I can model the dose per cell over time. And if I do that, then here you can see this is our experimental data again, the death versus time response. And now the solid lines here are our model, just with knowledge of the biological variability distribution and our finite reservoir delivery dynamic. Okay, so this experiment is the standard type of experiment we would do where we have a fixed volume and different concentrations. We can do this in a different way where we can have a fixed concentration but change the volume. And I think this is particularly important when we think about standardization and reference. Here's a 6 nanomolar concentration, 50 microliters of solution, and you can see the cell death at 24 hours, somewhere close to 15%. If I double my volume, I've doubled the number of particles in my reservoir, I double my cell death. And this is extremely important because I've looked in the literature, and quite often the literature will quote you a concentration, but it won't tell you what the volume used was. And so again, coming back to trying to get robust standards and robust references, you have to know particle number. And concentration isn't enough for that. You have to know concentration and volume, then you can understand what's going on. And once you've done the experiment, I think this looks very obvious, but I have gone back to literature and search, and I can quite easily find papers where I wouldn't know the volume used. That it would be just a concentration quote. Okay, in the interest of time, I'm going to skip this and just come to the final couple of slides. And so I've been talking through this in terms of a dose over time and the variability due to the concentration and changes in volume. So what I can do here is now go to a dose response where instead of plotting the exposure concentration, I actually plot the cumulative dose, this ND, that is in the cells at any given time. So through this equation, I know at any point t, any time t, I know the cumulative dose. And if I do that, I get this very nice global response curve. Okay, so now this is percentage cell death as a function of the accumulated particle number per cell. And you can see now my data at different concentrations collapses down onto that single global curve. And what we're doing here is we can either plot out that curve through time. I increase my particle number just by waiting longer. I have more delivered particles. And so the red line there is the highest concentration. And you can see over time, from one single concentration dose, I can get a full dose response curve. Or, as we would normally do in assays, I can measure at fixed time point, here at 24 hours, and different concentrations, and just look at the cumulative number at the end of some time frame. The other thing I can do is, because I know this is saturated here, I know the volume and the concentration, so I know the total number of particles delivered. And so in this case, we can actually then get an idea of the toxicity. So ND50 is the number of particles that gives me 50% death in the population. And I can calibrate that to actual physical number of particles per micron squared onto the cell. Okay, so let me finish off with this and make the following points. Concentration, as I said at the start, is an exposure metric. It's not a dose, and I would argue that if we really want to get standardization and understand each other's measurements and other people's measurements reported in the literature, we must have dose as particle number per cell, or at least total number of particles in the experiment. And then what I've done to summarize this is, from my point of view, what I might consider points of good practice. So as I've said here, using number rather than concentration. Using multiple time point measurements, I would strongly endorse. And also using variable volumes, just to check that in your experiment you're not having a supply limitation due to limited number of particles. And as I've just been showing, a cumulative particle number dose versus response would be our chosen metric. And if you look at all those and say what might be a minimum information set that we should all be publishing, and I stress this is just the minimum information set just to look at dose, okay? Response and biological interaction, biological mechanism, we would want to know a whole load of other things, but just to fully calibrate the dose of the system, obviously we need to know size, but we need to know concentration and the volume used, and actually what we also need to know is the dimensions of the assay well, because I can take the same volume, same concentration, put it in a different geometry well, and also get different results. Okay, my last slide, let me finish up. I don't know about you, but quite often in science, I have some ideas, I get excited about them, go, oh yeah, great idea, and I usually find someone's been there before me, okay? And so here, sure enough, over 75 years ago, Chester Bliss published this paper where he talks about doing toxicity assays in terms of a time response rather than a concentration response. So you can see here, calculation of time mortality. He was doing it primarily because he makes this observation that when you do a standard dose response curve, you're taking a different set of organisms, a different set of cells, each time you do a different concentration. So if you're a purist, that effectively means you're measuring the response on unrelated populations. And so if you do a time response curve, it's a single population of cells, and you're measuring that response over time. It's always on the same population measurement. For nanoparticles, I would add to this that yes, we should measure over some time response, but also because of the finite supply, even those observations at successive times are subject to change as the changing dynamic of the effective available number of particles changes the effective dose. So let me finish with my acknowledgements. As everyone who comes to speak, there are many, many people back home doing the work. So thanks to all my colleagues and students in Swansea, the people in Leeds who do the electron microscopy for us, and to wider collaborators at the Broad Institute in Boston and the Methodist Hospital Research Institute in Houston. And finally, thank UK sponsors, EPSRC, who have funded this work under this grant. So thank you very much for your attention. Thank you very much.
Thank you, Huw. We are already 1 minute late, but in the interest of the very fantastic talks, can we take one question? Yes, please.
Well, actually, I was going to press two questions into one question. Firstly, do dead cells re-release particles? So is your dose precise? And secondly, do you have any concepts of the doses in a dead cell at the point of death? You talk about susceptibility, but have you any proof that the actual dose that kills the cell is different between cells?
So in terms of particles getting re-released when the cells die, then obviously they will, although I don't know enough biology, will they be released still membrane bound from the vesicles? Will the vesicles be re-released, or will they actually fragment and get free particles again? So that could happen, definitely. In terms of the dose actually at the point of cell death, what we have is a measured fluorescence intensity at the point of death. So although I don't know directly the dose, I do know that variation in biological profile matches my fluorescence intensity at the point of cell death. So I'm fairly confident that we have an indirect measure of particle quantity at death.
Thank you. I think we must move on. I'm